Hello, my name's Olivia J. Kai. I'm a senior majoring in environmental science at Westfield State, and this semester for my senior capstone project, I focused on improving pollinator habitat restoration techniques in Western Massachusetts. So, pollinators are an essential factor to environmental and human health, responsible for the ecosystem service pollination. Despite their high ecological value, pollinator species are rapidly declining worldwide due to factors such as disease, exposure to pesticides, habitat loss, fragmentation, and more. Determining the relationships between pollinator species richness and abundance and different environmental factors is important to improving pollinator habitat restoration techniques. The primary goal of this project was to expand restoration methods regarding pollinators in Western Massachusetts. To do so, I decided that I needed to determine the success of the Spring 2022 Pollinator Habitat Restoration Project, and I needed to assess the factors that contribute to variations in pollinator richness and abundance. This semester, I made a comparison of the Spring 2022 data and Fall 2022 data. There were 3 separate locations at Westfield State University that I observed to determine the factors that contribute to pollinator richness and abundance, and I used 2 methods for observation: visual observations and blue vein trap observations. I hypothesized that pollinator abundance would be higher in areas with a high pollinator flower bloom abundance. I also expected that pollinator species richness would be higher in areas with a wider variety of pollinator flower species. So, here are some of my methods for determining the success of the spring restoration project. Pollinator plant survival rate was determined in each of the spring 2022 restoration locations. There were 7 locations total. 6 were located in the floodplain area, kind of down by the research area by Route 20, and 1 was an urban upland plot, and that plot was the pollinator garden on campus. Pollinator plants within the plots were identified and marked with a green ribbon. Weed removal via hand pulling occurred in the pollinator garden in 3 separate sessions. So if you look at that top photo, that's the pollinator garden before any weed pulling happened, and the bottom photo is the pollinator garden after weed pulling happened. As you can see, after the weed pulling, the garden was pretty bare, so most of the plants that we planted during the spring 2022 semester, it doesn't look like they were really showing up due to the abundance of weeds. This semester I used 2 main methods for my observations, one being blue vein trap observations and the other one being visual observations. I put my blue vein traps in 3 different locations this semester. One was the reference area, which had a very low abundance of pollinator flowers. The reference area was located behind Horace Mann at Westfield State. The pollinator garden was another of my locations. It had some pollinator flowers. That is located kind of by the Science and Innovation Center. And the community garden. The community garden is also behind Horace Mann, or next to Horace Mann actually, and it has an estimated 10 times the number of pollinator flowers present when compared to the pollinator garden. So the community garden was the most abundant in pollinator flower abundance, bloom abundance. If you take a look at the pictures to the left of the screen, you can see the top left photo is the reference area. The top right photo is the pollinator garden, and the bottom photo is the community garden. I removed the samples from the blue vein traps every 2 to 3 weeks and later identified them in the lab. I also did multiple visual observations in the 3 locations as weather allowed. The observations were 5 minutes long. There was 1 to 3 individual observations done in each location every 1 to 2 weeks. Observations were done in an estimated 5x5 imaginary square that was placed in a new location within the subject areas each time, and data such as weather, date, time, bloom abundance, pollinator species preference, pollinator species plant preference, pollinator richness, and pollinator abundance were collected. Here are some of my results for determining the success of the Spring 2022 restoration project. The restoration efforts done in the floodplain areas during the Spring 2022 project were 100% unsuccessful. If you look at the 2 tables towards the top right of the screen, you can see that the lower floodplain area and the upper floodplain area both had 100% death rates for all of the plants that we had planted during the spring 2022 semester. The pollinator garden, however, was slightly more successful. An estimated 25 pollinator plants out of 125 survived, giving us a 20% survival rate. If you check out the table at the bottom right of my screen, you can see the urban upland pollinator habitat table. 125 plants were planted, 80% of them died. So that leaves us with a 20% survival rate, and this 20% survival rate deems the spring 2022 project somewhat successful. So for my blue vein trap results, there's very little variation in pollinator species richness between the 3 locations that have very different pollinator bloom abundances. So if you look here, you can see that this, my y-axis represents the number of pollinators, and my x-axis represents the locations that I observed this semester. The blue bars represent pollinator species richness, and the orange bars represent pollinator species abundance. So you can see when you look at these blue bars in my 3 locations, they're very, very similar. There's not much variation. There is slightly more variation, however, between pollinator abundance between the 3 locations, but the variation is not as significant as I hypothesized. So if you check this out right here, you can see the difference between the 3 areas. They're pretty similar. The least abundance that was found was in the community garden. I found that very surprising, seeing how there was a pretty wide variety of pollinator plant species in the area. And my reference area had no pollinator plant species, so it's very odd that the abundance is as high as it was for the reference area. So I found that pollinator species richness and abundance decreased later in the year as temperature decreased, which suggests that temperature had an influence on species richness and abundance. If you look at the table on the bottom right of the screen right here, you can see that after November 7th to the 14th, for almost all the locations, richness and abundance dropped significantly, but for some reason in the reference area it was very oddly high. During my visual observations, I noticed that there were no pollinators here throughout the semester, so the variation in this data suggests to me that the data on species abundance collected via blue vein traps is not as accurate as when collected using visual observations. Doing my visual observations, I found that different pollinator plant species attract different species of pollinators. I noticed that there was a relationship between European honeybees and bumblebees being attracted to the purple aster, which you can see at the bottom left of the screen. And I also found that European honeybees are attracted to perennial sow thistle, which is at the top left of the screen. I also found that once pollinator blooms started to wilt and bloom abundance decreased, pollinator abundance also decreased. So in locations that were once abundant with pollinators and pollinator flowers, once the blooms wilted, the pollinators were also gone. In conclusion, the restoration project during the spring 2022 semester was 100% unsuccessful in floodplain locations. However, it was somewhat successful in the pollinator garden. Some methods are better than others for determining specific factors related to pollinators. Visual observations give a more accurate description of the pollinator species abundance of an area, and blue vein trap observations give a more accurate description of the species richness of an area. Pollinator bloom abundance is an important factor to pollinator habitat restoration. However, focusing on specific species needs rather than pollinator needs in general will improve restoration techniques. So we should focus on a specific species and their needs rather than pollinator restoration in general. Here in this picture, you can see some of the pollinators that I caught using the blue vein traps. I preserved them and pinned them and donated them to the environmental science department to be used for research purposes. So that was a fun part of my project, I would like to point out. Thank you for watching my presentation. If you have any questions or comments, please feel free to email me at agj.ek@westfield.ma.edu, and I'll gladly respond to any comments or questions. Thanks.